Thank you, everyone, for coming to this talk. I'm a student at the University of Notre Dame from the Farig Laboratory, and our key interest is in using genomics tools as a basis for understanding mechanism of action of drugs and drug resistance. And to give a small background, malaria is a disease really that has a huge economic burden in the world, primarily affecting sub-Saharan Africa. It's caused by the parasite Plasmodium falciparum, which has a life cycle stage in the mosquito as well as in the human. There's currently no vaccine available, and drug resistance is really prevalent. So we are motivated to use genomics tools as a basis for understanding drug mechanism of action. We know that gene expression is really a good measure of real-time responses of cellular systems to perturbations. And gene expression has been used successfully in the discovery of mode of action of anti-cancer drugs. But Plasmodium is really a unique organism when it comes to gene expression. So we know that the transcriptome of this parasite is highly developmentally regulated. So if you look at different developmental stages, one stage called ring, another trophozoite, another schizont, you see that genes show transient changes throughout the developmental cycle. But at the same time, we know that this organism has a very few set of transcription regulatory genes, and there are also very few DNA elements that have been discovered. And transcriptional variation is actually thought to show very little variation across strains, and also to show very little variation under different environmental perturbations. And so our interest is to develop tools that can really mine transcriptional data for very subtle patterns and how they relate to drug mechanism of action. And conceptually, the communication between a cell's exterior and interior is mediated through signal transduction pathways. And so we know that when we expose a cell to a drug affecting a given pathway, that leads to activation of transcription factors that eventually leads to changes in gene expression. And we expect that these changes in gene expression should be related across drugs that have similar mechanisms of action. But really, many times we don't know what's happening inside the cell. And so we have no way of guaranteeing that there's a one-to-one mapping between the drug mechanism of action and the observed transcriptional changes. And so to test this idea, we have started out with a small pilot of 10 drugs whose mode of action is known. And we have selected pairs of drugs targeting different pathways. And then we expose parasite cultures at one developmental time point. This parasite has 48 hours of life cycle. So we target one developmental time point for 2 hours. And then we measure gene expression using an exon array we recently designed. And then one of the things, of course, that emerges is that drugs that have related mechanisms of action show similar transcriptional responses. Of course, in reality, most drugs have multiple modes of action, and mostly there's only a focus on maybe the primary mode. Even so, if we have this knowledge for tons of drugs, then in future we can query this kind of database with the transcriptional profiles obtained from novel perturbations to understand modes of action of new compounds. But really, biological responses are complex, and because there's not a one-to-one mapping between each pathway and each gene, because one pathway might have several genes, so even drugs that target the same pathways might lead to differential changes in gene expression. And so it's important to look at biological data in terms of the pathways that are affected by the drugs. And so to do that, we can represent the cellular response, transcriptional response to drugs into pathways as a binary vector. So in this binary vector, we have a 1 if the pathway is activated by the drug, and 0 if the pathway is not activated. And so just to give an example of these two drugs here, we can see for each case whether the transitionally induced genes involved in that particular pathway. And so the power of doing that is that we begin to see relationships between drugs on the basis of the biological processes they affect. So we can see that there are biological processes that are affected by multiple drugs, but you can also see that there are some biological processes that are uniquely affected by only certain kinds of drugs. And to look at this in a different way, we can transform that into a network in which two drugs are connected if the patterns of pathways they affect show a very high amount of overlap. And the power of this is that we begin to see complex relationships between drugs. So for instance, this drug artemisinin, the key antimalarial, whose mode of action is not known, we see that it's associated with this group of drugs that affect heme polymerization and another drug that affects mitochondrion. So maybe this reflects a multiple mode of action for that particular drug. And we can see for drugs such as 5-fluorouracil, which affects DNA damage, but is also associated with the folate pathway. So you can see this complex relationship between drugs emerge. The other thing, there are also novel associations that we see in our data. So for instance, 5-fluorouracil and chloroquine. Chloroquine affects heme polymerization, but there's no really known association between the two drugs. And so kind of to generate an hypothesis of how that could be happening, we decided to use literature. And basically the idea is that if a drug targets a biological pathway, it's very highly likely that papers talking about that drug will use similar words like papers talking about the pathway targeted by the drug. And this includes cases in which there's not an explicit relationship between the pathway and the drug. And so to test that idea, we wrote a program that goes into PubMed and downloads abstracts for each pathway and for each drug. And so from this, we can try to predict what is the pathway target for 5-fluorouracil based on the word frequency in papers. And so based on that, we correctly predict that chloroquine, for instance, is associated with the hemozoin formation. And we found out that both chloroquine and 5-fluorouracil are highly associated with apoptosis. So maybe that is a possible mode of action. But even more importantly, if these gene expression responses are specific, what is the association between functional groups of a drug and the observed gene expression responses? And the key idea behind that is how do cells manage to respond to potentially millions of chemical molecules? It therefore means that cells might be recognizing some fundamental substructures in the chemical rather than the whole chemical as a whole. And so if that is true, then we can begin to map the association between the occurrence of specific chemical substructures and gene expression changes observed in data. And so one way to do that is to take each drug and split the drug into chemical subcomponents or substructures. And so each drug is represented by a binary vector in which you have a 1 if the chemical substructure is present and a 0 if the chemical substructure is absent. And so we can begin to mine the patterns between the occurrence of the substructure and whether the drug affected particular pathways. And so this allows us to find rules, for example, shown here, that the presence of a certain number of aromatic rings increases the likelihood that the drug will affect ATP synthesis, at least at the gene expression level. And so if we can mine those rules, then we can be able to conduct novel predictions where we can take new drugs such as rotenone and pass it through the model to see what the model will predict. So here the model says that rotenone would inhibit ATP synthesis, which is what is expected. And it also predicts correctly that ubiquinone, which is a natural electron which is a natural electron acceptor in the mitochondria, would not be an inhibitor. And so in conclusion, we think that even in Plasmodium, where gene expression responses are very subtle, they still carry some patterns that can be used to associate relationships between drugs, but also to mine complex relationships between drugs, because most drugs affect more than one pathway. And lastly, we think that this is also powerful in the sense of trying to associate chemical components with basic biological responses in cells. And just one last thing. So I'm really thinking about malaria and the possibility of using collaborative competitions as a way of looking at this disease, because really malaria is highly understudied when it comes to systems biology, and I think there's really need for new talent in this field to explore the parasite and to see how genomics and systems biology can actually help to find solutions. And I would like to acknowledge my advisor, Dr. Michael Farig, who has been very supportive, and an undergraduate student in our lab who has been helping me with the experiments. And to you all, thank you. So on the first question, one way to convert the transcriptional responses into binary fingerprints is just to take the differentially expressed genes, for instance, and perform Go ontology analysis. And so if a particular pathway is enriched beyond a certain threshold, you just encode it by 1, and if it's not enriched by 0. But you can also use things like gene-scent enrichment analysis to avoid like the effect of statistical thresholds. On the second question, to convert the drugs into binary vectors, we use standard chemical fingerprints, so like the PubChem fingerprints. So those are standards. We just use the standard ones. But of course, it's possible that we can fine-tune it, because we might know that there are common functional groups in drugs that are not necessarily explicit in the standard fingerprints, so we can incorporate those as well.